Hello, everyone. I am Professor Daisuke Kihara. I'm going to present molecular software bioinformatics software for drug screening and protein structure modeling. Proteins are fundamental working molecule in a cell. Knowing the 3D structures of proteins are very important for drug screening, drug design, and designing biomaterials of proteins. Nowadays, computational methods can accurately model and doing better drug screening by using AI-based methods, including deep learning. Our group will provide software, modeling service, and consultation toward the bioinformatics needs. The application of our software include structure modeling for structure-based drug design, designing protein biomaterials, and drug design using cryo-electron microscope data. Using bioinformatics is a growing market. Bioinformatics itself is growing to $14 billion in 3 years. And one of the largest applications, drug discovery, is of course growing very quickly to over $85 billion. And computational modeling in protein engineering is getting more and more popular, and that market is also growing. And one application in cryo-EM is rapidly growing market. Our technology will provide unified protein bioinformatics software powered by deep learning. Our tool have been rigorously benchmarked and compared with other methods in peer-reviewed journals. And we have also a very strong track record in competitions by objective assessment in Capri docking, CAFA protein function prediction, and CASP protein structure prediction competitions. Our methods are now using state-of-the-art deep learning methods. So I'll quickly show you the structure prediction methods and also the drug discovery in cryo-EM and gene function annotation. So this is an overview of our methods for structure prediction and protein complex modeling. Our structure prediction methods uses deep learning, state-of-the-art architecture, and our docking methods, Lizard and Dove, uses innovative protein surface representation and also deep learning for selecting good models out of the wrong models. Here I'm showing the recent ranking of protein docking methods in Capri protein docking competition. Our team is ranked number 4 here, and actually this is the best group in the US. This is our drug screening software, PL Patch Surfer. Advantage of our method is that using our improved structure prediction methods, this method's Patch Surfer can use computational models of proteins as target proteins. So that can increase the target protein space drastically because you don't need to have the solved structures as target. In comparison with other methods, the TBM, this is a template-based modeling methods results, does not drop significantly compared to the situation when you have the experimental data, but other methods significantly drops if you use computational methods. We have expanded our area to cryo-electron microscope images. Our methods have been published in high-profile journals, including Nature Communication and Nature Methods. By using deep learning, we can see the structure information in even lower resolution images or from cryo-EM, which was not possible before. This is about our function prediction. In the function prediction, our team was ranked number 1 in human protein function annotation. The next step is, by presenting this, I'm looking for industry partnership for collaboration and developing further. And I'm also looking for industry customers who need specific modeling services and funding for packaging software for dissemination. Please contact me. You can find more information in our lab website. Thank you very much.
Hi, I'm Craig Sweet, a postdoctoral researcher at Purdue University in Dr. David Thompson's lab. I'll be describing our novel strategy for protein purification using organic solvent extraction. Since the advent of modern medicine, small molecule drugs, such as antibiotics, have completely changed the world. However, the world is once again rapidly changing through the successful commercialization of biological drugs. These can take place in many forms, including antibodies, recombinant proteins, and as all of us know, vaccines. As you can see in 2010, biologics made up only 20% of sales. However, out of the top 100 drugs, they made up nearly 34%. Currently, biologics make up nearly 30% of sales and 50% of the top 100 drugs. Beyond this, these biologics also generate nearly $1 billion more in sales on average when compared to their small molecule counterparts. Due to this large expansion in biologics, the extremely important market of protein purification is rapidly growing to keep up. However, innovation in the protein purification market always lags behind and is not nearly as scalable and cost efficient as in the production of small molecules. Very complex systems are routinely required to produce biological drugs, such as recombinant proteins. Thus, in most cases, multiple techniques are required to be used. In 2019, the US protein purification market was nearly $6 billion. With its strong growth, it's expected to reach over $11 billion by 2027. By looking at a typical purification for an antibody, 4 expensive and slow, inefficient steps are needed to effectively remove the 3 key contaminants typically found in biological systems. The contaminants include host cell proteins, or all of the other proteins that are normally present in the expression system, nucleic acids, which are frequently co-purified using traditional techniques, and most importantly, when the cost-efficient bacterial system is used, special care must be taken to remove lipopolysaccharides, or LPS, which is an integral membrane component found in high concentrations in the host. All 3 of these contaminants can cause extremely dangerous effects if not completely removed to their acceptable levels, and may hinder scientific research. During my work as a PhD student, I worked extensively with a unique class of proteins called elastin-like polypeptides. These proteins were derived from a sequence of elastin, or skin protein, and have shown great promise in the field of drug delivery. In our pursuit to improve our purification method, we developed a novel procedure which was able to effectively purify our ELP directly from cells extremely efficiently by simply mixing a bacterial pellet with a polar organic solvent. Our protein of interest was selectively solubilized, while other host cell proteins were effectively removed. A precipitation is then done to remove the organic solvent, and our elastin-like polypeptide can once again be dissolved in water. Beyond its ability to selectively purify our protein and not other host cell proteins, it also removed nucleic acids and the notoriously difficult lipopolysaccharide below our limits of detection. Then, through a collaboration, we effectively purified a fusion protein for macular degeneration and saw once again it was successfully purified using this method and maintained its activity. This has inspired the development of numerous other effective purification schemes for elastin-like polypeptides and ELP fusion proteins, which maintained this unique property. We have currently shown success with numerous proteins and seek to implement our purification with other proteins of interest. With these advancements, we are looking for partners across pharma and biotech who seek to reduce operating costs and turnaround requirements for protein purification. While the market is large for development towards human health, this is not our sole focus. Proteins are becoming a larger product across other industries, such as bioadhesives and bio-inspired materials. Thank you.
Okay, hi everyone. My name is Dongwoo Kim. I'm CEO of Denkoda LLC. So our first minimum viable product is early detection of prostate cancer using our DNA-based sensing technology. So prostate cancer diagnostics are generally using PSA test kit, but this test kit has only 30% accuracies. So you have to do unnecessary follow-up tests such as biopsy and some ultrasound, more expensive tests. So you have to spend more than $2.3 billion per year is wasted. So try solve these problems. So using our technology, so you can improve accuracy of this prostate cancer testing up to 90%. And then we can save $2.1 billion per year. So current market is total addressable market for prostate cancer diagnostic is $19.3 billion. And among them, service addressable market is US PSA testing market, $2.1 billion. And then our target market is $9 million. That's calculated from total cost of PS testing spending IU, Indiana University Health System in Indiana. So this is our early target market using our product. So our technology using DNA linked activity probe. So you can adding active site probe, in this case, prostate cancer biomarker, we can react with that prostate cancer biomarker using our DNA linked activity probe. And then you can pull down by that biomarker a specific antibody. And then we can anneal our barcode DNA to this DNA linked activity probe. So we can detect amount of this active protein amount by qPCR or DNA sequencing. So we published this data at Chem Communication in 2017. And then basically you can see the cartoon here. You can attach DNA barcode here, red things. So you can recognize this active enzyme pulled down by antibody. You can detect this active protein by abundance of DNA. So if you have more enzymes there, you can have a more DNA recover from immunoprecipitation. So compared to other competitors in the market, like such as Walgreens, you can go CVS, you can find this PSA testing kit. So this testing kit, you can do blood sample. You can take out blood and then you do some testing, but accuracy is not that great, as I mentioned about, only 30%. Time to report 1 to 2 weeks. The sample size, you need a lot of blood for testing. But that's why the other companies, OPKO and Beckman, they have a new developed assay to detect these prostate cancer biomarkers. So they're using, in OPKO case, a urine, and then Beckman's case, blood. But in our case, we don't have any sample restrictions. We can use either blood and urine. It's fine. Accuracy is more than 90%. Then turnaround time is 2 to 3 days. And the sample size, you don't need that much as blood to analyze whether or not you have prostate cancer. So we are asking the industry partnership, we can collaborate with them. And then also clinical prostate cancer research, we can provide our technology to study about prostate cancer biomarker function. Also we can identify new biomarkers. So we also ask some investors, we can talk about our technology, the possible licensing technology or collaboration, all these possibilities. Thank you very much.
Hi, my name is Liz Top, and this presentation is entitled Human Calcitonin Derivatives with Improved Stability in Solution. These are drugs essentially to treat osteoporosis, and there's a real market need for such drugs. There are about 10 million Americans in the US that have osteoporosis, and 34 million more people are at risk. We have drug treatments for osteoporosis now, and they include bisphosphonates like Fosamax, salmon calcitonin, and some monoclonal antibodies like denosumab. You might be interested to learn that we're using fish calcitonin to treat human osteoporosis. And the reason for that is that human calcitonin, the human version of this peptide hormone, is unstable and rapidly forms amyloid fibrils. Because it's so unstable, it doesn't make a very good drug product because we can't keep it stable on the shelf. The problem with salmon calcitonin, on the other hand, is that it has some side effects associated with it. And so we think there's a need for a stable form of human calcitonin. Just a little more about the market. Osteoporosis is kind of a big deal and getting bigger. The global osteoporosis drug market is expected to reach nearly 11 billion by 2026 with a 4.4% CAGR. In the US, there's a huge economic burden, 17 billion already back in 2005. And if you're over 60 and you break a hip, there's a 20% of those people who do that will die within 6 to 12 months, and 50% will lose their independence, their ability to live independently. And while, as I said, the bisphosphonates are helpful for this, not everyone can take bisphosphonates. And in the little box on the right, I especially want to draw your attention to that projected increase in hip fractures, which is a big part of what we're trying to prevent. Up to 300% increase projected by 2050 in men, and a nearly 250% increase in women. So we think we have a way to help with this. We've developed a phosphorylated form of human calcitonin. And what you see there in the kind of text up on the upper right, the kind of cryptic looking text, for those of you who don't recognize it, is the amino acid sequence for human calcitonin, essentially a shorthand version of its structure. And the red circle around the THR identifies the site that we're phosphorylating. So as you see in the bulleted list, phosphorylation of threonine 13 completely shuts down the fibrillation of this hormone. And so we can make a stable solution form of human calcitonin when it's been phosphorylated. The follow-on good news after that is that in the body, phosphatase enzymes will clip off the phosphate group that we put on there, regenerating the native human calcitonin. So the patient is seeing a drug that was stabilized by a phosphate group when it was in the formulation, but once it's in the patient, the phosphate group comes off and the patient gets the native hormone. Those of you who know the pharma space will recognize this as a prodrug strategy. And it's a type of strategy that we've already used successfully for another badly behaving peptide hormone called glucagon. So just to talk about the calcitonin specifically and why we think this is a good strategy, you see three different choices on the left there: salmon calcitonin, human calcitonin, and our suggestion for phosphorylated human calcitonin. We think all of them are effective. Salmon calcitonin is the marketed drug product of choice. Human calcitonin is also out there, but hasn't really gotten a fair shake, we think, because of its instability. Because of that, we think phospho-human calcitonin will be effective too, although we haven't used it in humans. Because it reverts to native human calcitonin, we think it'll work just as well. Salmon calcitonin is pretty stable. Phospho-human calcitonin is pretty stable, but of course the big red X in the center there is that human calcitonin is not. What we're also trying to get away from here is some of the side effects associated with salmon calcitonin. Some of them are not so bad, but there's higher incidences of cancer in some patients receiving calcitonin over salmon calcitonin over the long term. And we don't really have as much data on human calcitonin or phospho-human calcitonin, but we think they deserve a fair shot. And in terms of being an effective treatment that may have a better side effect profile, and that's part of the shot is what we think the phosphorylation will help us offer. So what are we up to? What do we want for next steps? We're looking for collaborators who have clinical and preclinical experience in the osteoporosis space. I'm a peptide protein formulation person, and I don't have expertise in that area. We're also looking for funding always, because we'd like to develop a more in-depth portfolio to further progress this technology and show what it can do. And you see some of the things we're thinking about there. And finally, we would also like to ultimately license to pharma or biopharma so that they can market this interesting new drug product and get it out to patients. Thanks for your attention. I look forward to your questions.
Good day, everyone. My name is Prakash Nalthambi. I'm the Associate Director of Research for the Institute of Precision Health at the University of Notre Dame. Concurrently, I'm also an Assistant Research Professor in the Bioengineering Department, and my lab focuses on tackling real-world problems by trying to translate innovative technology from the lab into the market. So keeping in that spirit, we have been looking at new solutions to tackle the antibiotic-resistant crisis. And so we come up with a new class of antibiotic-free antimicrobials, which we have termed loosely as the phage-mimicking nanoparticles. So as far as market need goes, according to a WHO report, by the year 2050, antibiotic-resistant bacterial infections will kill more people than all cancers combined. That's how dire the scenario is right now. It's only now that industry and governments are waking up to this problem. As you can clearly see from the number of antibiotic-resistant infections each year, which numbers in millions, and then you have like tens and thousands of deaths from the same multi-drug resistant bacterial strains. Why is there such a big problem? The problem really is because whenever we come up with a new antibiotic, it's not a question of if, but it's a question of when that bacteria starts developing resistance to this and then passes that on to other bacteria. So new antibiotics are very strong. They have horrible side effects. And nowadays, they're used as last resort methods if existing antibiotic cocktails fail to have any effect. Alternative antibacterial substances such as silver nanoparticles or silver ions are effective in certain circumstances, but they have inconsistent therapeutic outcomes, and they also suffer from biocompatibility issues. So there's definitely a need for better means of controlling these multi-drug resistant bacteria. And there's definitely a need to look at non-conventional sources and not just keep looking for the next new antibiotic. As far as the target markets go, it's very broad. It can be anything far right, such as the animal health, to something in the consumer sector, such as acne treatment and cosmetics, to improve the shelf life of cosmetics. It also has wide use in something like the dental industry. They have oral rinses that will keep the buccal cavity free of pathogens, prevent infections after a dental procedure. And of course, there's anti-infectives and antimicrobial coatings on medical equipment. And the numbers are here, and these are easily accessible, so I'm not going to go into details about this. As far as our technology itself, like I said, it's phage mimicking because it has a silica core and it has gold silver nanoalloys studded on top, which kind of mimics the structure of bacteriophage with its protein spikes. And each one of these is antibacterial, but we can also further enhance them by putting polycationic polymers or do bioinformatic mining and look at antibacterial peptides, and then modify them to be tagged on the gold silver nanoalloys as well. So the bottom line is we tested this against 3 gram-positive, including MRSA, and 1 gram-negative, which is Pseudomonas, multi-drug resistant Pseudomonas. And in each case, we had nearly 100% kill rate. We had the same effect regardless of if we immobilize the particles or had them diffusing in solution. 100% biocompatibility with moral skin cell line, and it utilized a 1,000-fold lower concentration of the antibacterial peptides compared to current state of the art. If these peptides were used as is, we lowered that concentration by 1,000-fold, which means we're saving material costs. So again, to emphasize, it's antibiotic-free, We got positive market feedback on a need. There's an unmet need for antibiotic-free disease management from companies like Elanco and Merck, as well as USDA and NIFA. So what we're looking for, we're looking to accelerate the product translation out of the laboratory. We are at tier 3 to 4 right now. So we are looking at industry partnerships or any collaborations to validate our platform technology in vivo. And we believe this will allow us, this kind of partnerships and collaborations will allow us to pursue funding opportunities, private or government, which will then push it to TRL5 and above, which would be the next logical series of steps to take this from the lab into the marketplace. A quick comparison compared to other antibiotic manufacturers, ours is easily, meaning fast, meaning we can quickly pivot to match whatever the end user need is. It's a very strong antibiotic agent. It's broad spectrum, similar to all of these. Except Locus Biosciences is a phage company, so it's only specific to one pathogen, so it's not broad spectrum. So ours is broad spectrum, inexpensive, and very quick in our ability to pivot to meet the end user's challenge. With that, I'd like to thank you for your time, and please feel free to reach out to me or us if you have any questions about any of this technology. If you have any questions about licensing, if you have any questions about collaborations, we are very eager to hear your feedback, and we are also eager for very productive collaborations that might come out of this. Thank you.